This is a brief overview of pcmP, a projection comparison tool for single-cell RNA-seq. We will be looking at a live demo. You can look at it yourself using the shinyapps.io address given here. Source code is available in GitHub. This app works with Bioconductor 3.8, given a single-cell experiment that has some projections already placed in its reduced dim slot. We can compare projections, compare different facets of a single projection, and compute distinguishing signatures of groups of cells that we see in the display. This is what the shinyapps.io display looks like. And we want to think about the different controls that are available, the displays, the tabs that are available to look at different aspects of an exploration that we'll perform, think about how to interact with these plots, and finally how to save the work that we do as we work with the plots. This tab, About, is actually dynamic, and for different inputs it will give you a different report. In this case, we're being told that we are working with some data from the Tabula Muris project. We've limited the data to cells extracted from lung, and in fact we've even downsampled some of the cell types. Then the data have been filtered and corrected for batch effects after normalization. All that information can be recorded in a summarized experiment that you work with and is then propagated to this app through the About tab. The scatter plots are 4 panels, and every dot is a cell. There's a comparison of 2 different projection procedures in the top, or that's the intent, and the same projection procedure is looked at in different ways by columns. So here we have principal component 1 versus principal component 2, and here we have the third principal component versus the second. And we can use the controls to change that. So for example, we now are looking at the 4th principal component versus the 2nd, and we can then pop that up so that we now have the 3rd and the 4th. We can also change the projection on the right to t-SNE projection. It retains the same dimensions of each projection as before. We also color the cells using this stratify control, and we can look, for example, to see if there's a strong effect of the mouse identifier. And we see that in the first 2 t-SNEs perhaps there is. It's not quite as prominent in the principal components projection until you go down a bit. So these are the sorts of things that this tool is intended to help with, to get a sense for how sensitive a given interpretation is to the specific projection that's used, and to allow one to look through different features of these multiple projection schemes. Now, one of the things we'd like to do is work with the cell ontology class that was assigned to each one of these cells in the Tabula Muris project, and try to understand what gene expression has to tell us about the different cell types. So here's an example where we can see the T cells are fairly well separated from all the other cells. So we go and draw a box around them, and all the other cells that are not in the box are blurred. And so we can see that in different projections the T cells are more or less prominent, more or less mixed in with other cells. And once we've done that, we can then select the Cell Table tab, which will do a quick analysis of differential expression of the selected cells versus all other cells. and it's somewhat reassuring that we see these CD3 genes being differentially expressed in the selected population here, supposedly T cells. We can make another selection then. Let's see. In order to continue, we have to click on the panels to get back to selection mode. And now we'd like to isolate these alveolar macrophages. And we do that like that. Come back to the cell table, and it goes and finds the genes that are most differentially expressed. Again, the selection versus everything else. It's a very naive approach, but it does give us what seems to be some information about differences in gene expression distributions among these cells. Now this panel called Accum will tell us in one of the projections which cells have already been selected, and it also gives us some information on substructure within the clusters that we've chosen. These are biplots of a selected number of genes that are differentially expressed. To isolate this particular group, one is asking whether there are genes and possibly subgroups here which are distinguishable because of differences in expression. So that summarizes all the different tabs and interactions that we can take care of at this time. Once we've done an exercise of this sort, we can download the list of the differentially expressed genes to our disk, and also the lists of cells making up the different clusters. And we'll build additional tools in this package to help move things downstream. The GitHub repo is vjcitn/pcmp, and it starts out with a vignette that describes in more detail what's going on with the data set. And this is where you can actually install the software locally and use it on your own data.